Hey folks, I'm out here in the heart of the Salinas Valley at the organic research farm that I manage. And the video that you're going to see here is a little bit different than a lot of my previous videos. This is the first of a series of videos that we will be making to describe this really cool experiment that's in the background here. So this is an experiment that I'm doing with two collaborators, both Ph.D. students from Stanford University. And this is a study that I think is going to answer some really important questions about cover cropping and different approaches to cover cropping in the Salinas Valley. So this is the first of a series, and so I hope you'll stay tuned for some of the other videos on this. But this first one will just kind of describe the overview of the study, and then the subsequent ones that we'll make will describe some of our results and some of the different methods that we've used to collect our data. So hope you enjoy this. Take care. Winter cover crops like these can have many benefits. I want to focus on the ability of non-legume cover crops like rye to absorb or suck up leftover nitrogen from one season and then carry it over the winter so that it can be used by the next year's vegetable. That nitrate scavenging ability is the reason that cover crop credits were added to a new regulation called Ag Order 4.0 that's focused on protecting the groundwater from nitrogen pollution. But here's the question. What about non-legume cover crops that are planted in the late summer, terminated in the fall so that the beds can be made, and then left bare fallow over the winter for early season vegetable plantings? Could those also help to reduce nitrate leaching and benefit the next vegetable crop? If the answer to that question is yes, then perhaps growers could also get nitrogen scavenging credits for those fall-incorporated cover crops, not just for the winter cover crops. That, my friends, is the focus of the trial that my research colleagues Sierra and Anna and I are going to tell you about. It's a complicated trial, a really important one. It's got four main treatments. The first one is a winter cover crop of Merced rye. That's considered the best management practice to reduce nitrate leaching. And then on the other end of the scale, we've got a winter fallow with no cover crop. And then in between that, there are two fall-terminated cover crop treatments, both with rye, one that's grown for 60 days and the other that's grown for 90 days. We're comparing all these treatments in fields with lots of leftover nitrogen from the previous crops. And the reason that we're doing that is to make sure that there's a lot of leftover nitrogen that's at high risk of leaching into the groundwater. Now you might be wondering, why are we comparing a 60-day versus a 90-day fall-terminated cover crop? The reason for that is because even though they both take up about the same amount of nitrogen in their residue, the residue from the 60-day cover crop is younger and juicier and has a higher nitrogen concentration when it's terminated. And therefore, that 60-day cover crop will decompose faster and release the nitrogen into the soil more quickly, and that will possibly increase the risk of nitrate leaching compared with the tougher, older residue from the 90-day fall-terminated cover crop. So just to summarize, 3 of our treatments are in listed beds over the winter, and then we cultivate or flame those to keep them as weed-free as possible. And then only 1 of our treatments has a winter cover crop during that winter time. We're studying all these 4 treatments in a 2-year study at the USDA Agriculture Research Service organic farm that I manage, and we're now in the 2nd year of the trial. The trial's going really well. It's a lot of work, but we're getting good results. We're also doing a simpler version of the experiment in 2 on-farm trials in conventional fields. Anyway, we're measuring a whole bunch of different things that Sierra is going to tell you about next. Take it away, Sierra.
Thanks, Eric. So now that you have a bit of the motivation behind why we're doing this field trial and the setup, I'm going to show you how we're doing it. So like Eric said, we are quantifying the magnitude of various nitrogen loss pathways relevant to each treatment in order to determine how well fall cover crops reduce nitrogen leaching compared to winter cover crops. In order to do this, we have various data collection techniques that we use in the field and then analyze back in the lab. It is important to quantify how much nitrogen and carbon are actually going onto the field as cover crop residue. We do this by measuring the biomass carbon and nitrogen content at the time of termination. We collect biomass samples, track the peak's growth stages, and carbon to nitrogen ratio throughout the growth period of the fall and winter cover crops. Since we are interested in how environmental factors such as soil temperature and moisture impact the decomposition of fall versus winter cover crops, each plot in our field trial has a set of soil sensors that capture temperature and moisture at the soil surface, 30 centimeters and 60 centimeters deep. Measurements are captured every 15 minutes and uploaded into data loggers that are connected to the cloud for easy remote monitoring. This data will help to inform the seasonal variability between treatments and also serve as predictor variables for estimating nitrogen movement throughout the soil profile. To track the nitrogen release patterns of these cover crop residues, we also sample soil to analyze the concentrations of plant-available nitrogen, which includes nitrate nitrogen and ammonium nitrogen, from 0 to 30 centimeters, which is considered to be the root zone, 30 to 60 centimeters, and 60 to 90 centimeters, which is assumed to be leachable nitrate. This allows us to see the uptake of nitrogen from the soil into the cover crop plant tissue, subsequent breakdown of cover crop residues, and movement of plant-available nitrogen down the soil profile. Once the winter cover crop treatment is terminated in March, we then planted romaine lettuce to understand the impact of each treatment on subsequent cash crop yields. We also collect runoff samples of water and test for nitrogen concentrations, take microbial samples that we analyze for denitrifier abundance, root samples for below-ground biomass estimates, and measure soil greenhouse gas emissions. Now that we've shown you what and how we are collecting, Anna is going to discuss the questions we are really interested in answering and how we hypothesize things will shake out based on a previous laboratory study and the first year of our field trial. Over to you, Anna.
Thank you, Sierra. I'm excited to share with you 3 takeaways from our completed soil laboratory incubation study and 3 overall questions that we are planning to answer with our cover crop project. During the summer and fall of 2023, we conducted a soil laboratory experiment over 112 days with rye cover crop residues of 4 different carbon to nitrogen ratios: 10:1, 14:1, 19:1, and 30:1. We used soil collected from our organic research field in Salinas, putting the soil in 3 temperatures to represent the range of soil temperatures at residue incorporation. Our main objective was to understand how much of the nitrogen in the plant tissue is released into the soil as inorganic nitrogen and is at risk of leaching. We have submitted our experiment and results as a peer-reviewed journal article, but in short, our 3 main takeaways are: first, that rye carbon to nitrogen ratio strongly predicts rates of nitrogen release from the plant material. Second, for higher carbon to nitrogen residue, for example 19 or 30:1, the soil temperature has more of an effect on nitrogen release versus nitrogen tie-up or immobilization. And third, the risk of nitrogen leaching from fall or winter rye declines as the carbon to nitrogen ratio increases. Since we are still processing a lot of data and still collecting samples, we won't draw any concrete conclusions just yet. However, we want to give you an idea of what type of conclusions that we will have once we finish the project. Analyzing our 4 types of samples that Sierra mentioned: soil, down to 90 centimeters, water, both runoff and leaching, greenhouse gases, carbon dioxide and nitrous oxide emissions, and plant biomass, rye and weeds. Our 3 overall research questions include: 1. How does the residue decomposition and nitrogen release patterns differ for fall and winter cover crops? 2. How does the risk for nitrate leaching and runoff compare between fall and winter cover crops? And finally, what are the optimal rye cover crop management solutions for minimizing nitrogen discharge or loss from the field? Thank you for listening, and we look forward to your questions.